Hello, everyone, and welcome to today's webinar titled Optimize Your PCR. This webinar is part of the 7th annual event in the Laboratory Automation Virtual Event Series, and I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Thermo Fisher Scientific. For more information about Thermo Fisher Scientific, go to www.thermofisher.com. So let's get started. Before we begin, I want to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find that live chat located on the left of your screen. You can also participate by submitting as many questions as you'd like during this presentation. To do so, simply type them into the Ask a Question box and click Submit. In addition to asking questions, we encourage you to participate in our 3 poll questions at the middle of the speaker's presentation. Click on the answer when the poll questions are displayed. And if you have any trouble seeing or hearing this presentation, just click on that help desk button located at the top of your screen within the navigation bar or from the lobby. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on that continuing education credits link located in the abstract window below the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Gabriel Alves, Market Development Manager at Thermo Fisher Scientific. For a complete biography of our speakers, please visit the Presenter tab from the menu at the left of your screen. Dr. Alves, welcome. You may now begin your presentation.
Hello, I'm Gabriel Alves, and it is a pleasure to be your host today for this webinar. Today's topic is about optimizing your PCR. So we will go through the individual steps of the PCR workflow and review their function and meaning. Along the way, we will mention common pitfalls of PCR and how to avoid or troubleshooting them. We are going to talk about advances in PCR, DNA template, DNA polymerases and buffers, designing ideal primers, choosing the right plastics, controls, thermal cycling, post-PCR workflows, and also resources. Have you ever wondered how scientists are able to amplify specific segments of DNA millions of times over? This answer lies in the polymerase chain reaction, or PCR, a technique that has revolutionized molecular biology since its invention in 1983. But PCR has come a long way since its early days. Back then, it was a laborious and time-consuming process that required vast amounts of space and materials. Fortunately, with the ability of DNA polymerases with hot start technology, universal annealing, and also high fidelity, as well as user-friendly and modern thermal cyclers with very flex block technology, Thermo Fisher Cloud, and precision plastics manufacturing, PCR has become faster, more efficient, and more accessible. In this webinar, we'll dive deeper into the world of PCR, exploring different types of DNA polymerases and their unique properties. We'll also take a closer look at the latest advances in thermal cycler technology and how they have made PCR more precise and reliable than ever before. Whether you're an experienced researcher or just starting in the field, this webinar is for you. We'll cover the fundamentals of PCR and explore some exciting applications of this powerful technique. Let's begin with a brief background. Polymerase chain reaction, or PCR, is a molecular biology method used to amplify or multiply DNA. PCR works even from a single DNA molecule, each amplification cycle consisting of a template, denaturation, primer annealing, and primer extension step. During each cycle, DNA is copied and increased twofold. For example, after the second amplification round, there will be 4 copies of the same DNA. After the 30th cycle, 10 to the power of 9 copies of the same DNA. It is a lot of the same DNA. So it is a really powerful technique for DNA analysis. So what's needed for a PCR? Overall, there are 2 parts. First, the setup of reaction. where all necessary components are added, such as the DNA template with the target sequence to be amplified after successful DNA isolation, of course, a DNA polymerase to amplify the new DNA strands, the primers specifying the target sequence to be amplified, a buffer providing optimal reaction conditions for the polymerase, and the NTPs, which are the building block of the PCR product. Then follows the second part, the thermal cycling with steps for denaturation, which is melting of the DNA template to separate into its individual strands, the annealing of the primers to the DNA template, and then the extension of the new DNA strand by the DNA polymerase to form the complement strand. These three steps are repeated up to 40 times in a cycle to achieve amplification of the target sequence. As it begins with the setup, we'll look first one by one at the components added. The first component that we'll have a closer look at is the template. The amount of template used for a PCR reaction can significantly impact your results. It is recommended to use a range of 0.01 to 1 nanogram of plasmid or phage DNA, 5 microliters of complementary DNA from reverse transcription reaction, or 5 to 250 nanograms of genomic DNA. It is important to consider the template amount because it can affect the presence of inhibitors and the efficiency of the reaction. When using complementary DNA, it is common practice to use it directly for the PCR without determining its concentration to avoid sample loss and transfer of inhibitors, including the RT enzyme. In PCR, even one molecule of DNA can potentially produce a high yield of product. But in reality, a minimum amount of template is needed due to less than ideal amplification efficiency and the possibility of unspecific product amplification. The question often asked is what the minimum amount of template required is, but it's also crucial to consider the maximum amount that can be used. Using too much template can lead to an unspecific amplification and even PCR inhibition. An exact quantification of DNA to be used as a template is therefore very important. In the example provided, the same template was used with the same setup and cycling conditions, but diluting the template 1 to 10 resulting in no unspecific amplification. So as you can see in this figure here, 10 micrograms. As you see in the image here, the 10 micrograms of DNA template, you are obtaining a lot of unspecific amplification. These bands do not look good. And by diluting using less DNA template, you see that now that the bands are not unspecific. So therefore, DNA template quantification is extremely important. When quantifying the template amount, measuring the absorption at 260 nanometers is a common practice. However, it's also recommended to measure a few more wavelengths to assess the quality of the template and check for any contaminants. Checking the absorption at 230 nanometers is important, and if the absorption 260 to 130 ratio is less than 1.8, that indicates contamination with organic compounds, sugar, or salts. The absorption 260 to 180 ratio can be used to check for protein contamination originating from enzymes or cells. Additionally, absorption at 330 nanometers can indicate light scattering by particles such as silica breakthrough from spin columns. Keeping an eye on these ratios can give good idea about the purity of the template, ensuring a successful PCR reaction. Besides contaminating substances that can inhibit the PCR or cause it to fail, the template itself can be the source of the problem. For example, GC-rich sequences or such with high degree of secondary structures can be quite challenging to amplify as the strands of such DNA are more difficult to separate and therefore less accessible for the DNA polymerase and its primers. To deal with this, you can add additives such as betaine, DMSO, formaldehyde, or glycerol to the reaction mix. This will destabilize the hydrogen bond bridges and enhance DNA denaturation. However, if additives like DMSO or formaldehyde are used, higher amount of DNA polymerase is needed because these substances have a negative effect on the enzyme. Also, with all such additives, the annealing temperature will need to be lowered. This is because the prime binding will also be affected and destabilized. There are products available in the market that have special GC enhancers to enhance amplification of such templates. For example, AccuPrime GC-rich DNA polymerase is designed to provide high-yield, high-specificity amplification of difficult-to-amplify templates, such as those with greater than 65% of GC content. Specific kits optimized for challenging sources of nucleic acid can be used for extraction and purification. Adding less templates to the reaction can reduce the amount of inhibitor used. Increasing the reaction volume can dilute inhibitors present. Adding BSA to the mix can bind to impurities and prevent interference. Finally, using more robust DNA polymerase like Invitrogen Platinum Superphi 2 DNA polymerase can help overcome inhibitors during the amplification. Speaking of DNA polymerase, this is another component of the PCR, and we'll take a closer look at them now. While the template is the blueprint, the DNA polymerase is the motor of the reaction doing the work. So it's a crucial component, and it's good to consider all available options. So what's our best option? Activities of a DNA polymerase that are important for PCR are: synthesis of new DNA strands relies on 5' to 3' polymerase activity of the enzyme. All DNA polymerases possess this activity. Another is 3' to 5' exonuclease activity, which removes misincorporated bases. DNA polymerases with this activity are commonly known as proofreading enzymes because it helps ensure sequence accuracy during amplification. Polymerase's error rate is often expressed as the number of incorrect nucleotides incorporated per total synthesized. An error rate of low-fidelity enzymes is about 1 to 10,000 nucleotides incorporated, so there's a big discrepancy there. If we're looking up the error rates or fidelity information for different DNA polymerases, it is important to keep in mind that there are different ways to measure fidelity, and the results can vary significantly. There are 3 common methods used to measure DNA polymerase fidelity. On the left column, an overview of colony screening assays. In the middle column, Seeger sequencing, and in the right column, next-generation sequencing. Colony screening assays and Seeger sequencing both require huge data sets to see statistically significant and reliable error rates of high-fidelity DNA polymerases. Nowadays, next-generation sequencing techniques provide an effective method to analyze large amount of data and provide more accurate quantification method. When you're looking up fidelity of different polymerases, it is important to note the method that was used for the measurements. Now this slide explains 2 additional very important properties of DNA polymerases: processivity and hot start. Processivity is the measure of a number of nucleotides incorporated by the enzyme in a single binding event until the enzyme disassociates from the template. It is also related to the enzyme's affinity and how tightly binds to the template. High processivity of enzyme contributes to faster DNA synthesis, higher inhibitor tolerance, and longer amplification. Hot start is the activation of enzyme activity at a higher temperature. Enzyme is inhibited at room temperature and becomes activated during the denaturation step of PCR. Hot start mechanism contributes to specificity of PCR results. Hot start and high fidelity are not mutually exclusive, meaning high fidelity PCR enzyme can be modified to possess a hot start feature. Hot start PCR enhances specificity and amplification by preventing amplification of non-specific targets from misprimed sequences, which can result in reduced sensitivity and decreased yields of intended targets. While standard or known hot start enzymes are active in a room temperature during reaction setup, hot start enzymes are typically modified to be inactive at room temperature by antibody, chemical, or affibody molecule and an aptamer. As you see in this image here, this DNA polymerase has several antibodies binding to it. Then you apply heat and those antibodies dissociate, and now this enzyme is activated and ready to do the amplification. So, after heat activation, the hot start enzyme becomes active when it needs to synthesize a new DNA strand. Using a hot start enzyme provides better specificity and yield compared to standard enzymes. And here's the comparison. These two wells here, they're from not hot start PCR, and you see that the hot start PCR enzymes, the bends, are way more apparent. So better yield and better specificity. Therefore, it's a better approach than setting up the reaction on ice, which can only lower the enzyme activity but cannot fully deactivate it. Now, if you're wondering how to choose between low fidelity and high fidelity PCR enzymes, here's a simple tip. If you just need to detect the target sequences by their presence or absence, a low fidelity enzyme like platinum 2-Taq enzyme will do. Typical applications of low fidelity PCR enzymes include high throughput PCR screening, mouse genotyping, sequence detection, and colony screening. Now, high-fidelity PCR preserves DNA sequence accuracy and is recommended for applications like cloning and subcloning, template generation for sequencing, mutagenesis experiments, and genetic variability studies. Platinum Superfy 2, which we'll cover more later, is an example of a high-fidelity PCR enzyme. Our latest platinum PCR enzymes are designed with these two key unique features. They are based on the platinum hot start technology, therefore they get their names. This technology is different from others because of the properties of the antibodies that fully inhibit the enzyme activity until the initial denaturation step. That means greater target yield and detection sensitivity, as well as enhanced benchtop stability. The second feature is the universal primer annealing. It allows you to circumvent tedious optimization steps for primer annealing because you can now use a universal annealing temperature of 60 degrees Celsius for different primer sets. It allows you to cycle different PCR assays on the same block, for example, co-cycling, which will be explained soon. The universal annealing feature is made possible by isostabilizing component of the innovative buffer, which helps with specific enhanced annealing of primers. Here are three key benefits of universal annealing feature. This example here shows melting temperatures ranging from 59 to 69 degrees. Number two, one extension time for multiple targets. Extension time of the longest amplicon can be used for shorter amplicons, up to 5 kilobases, without sacrificing PCR specificity enabled by the isostabilizing component. The figure at the bottom left shows PCR results of enzymes with or without the universal annealing feature. Look at the differences. Because of the first two features mentioned, different PCR assays can be cycled together on the same block. For example, co-cycling, which is the bottom right figure. Typically, with other enzymes, different PCR assays need to be run sequentially, which can take way more time. Our latest hot start enzyme is platinum 2-Taq hot start DNA polymerase. Its key benefits are PCR simplified with universal annealing, faster synthesis and higher inhibitor tolerance due to enzyme engineering, enhanced specificity, sensitivity, yield, and benchtop stability from its hot start technology, direct gel loading with its green master mix format. Here's some supporting data from platinum 2-Taq hot start DNA polymerase. Its fast cycling enables you to complete PCR in less than 40 minutes compared to other enzymes on the market. If you look at the PCR runtime here, this is our DNA polymerase, our platinum 2-Taq DNA polymerase, and you see that the cycle was completed, the PCR runtime was completed in less than 40 minutes. The top right figure shows enzymes tolerance to humic acid found in soil, hermine, which is red blood cell component, and xylan, which is a plant biopolymer. The bottom right figure shows enzyme sensitivity down to 16 picograms of humic genomic DNA with high specificity. Formulation of platinum 2 Taq hot start DNA polymerase and 2 times master mixes allows for amplification of versatile range of targets from AT-rich to GC-rich. A separate vial of platinum GC enhancer is provided for specific amplification and improved yields of targets with high GC content over 65%. Now, our latest high-fidelity enzyme is the platinum superfy 2 DNA polymerase. Its key benefits are its extremely high accuracy or fidelity measured by next-generation sequencing, simplifying PCR optimization with primary annealing and GC-rich amplification, improved sensitivity, inhibitor tolerance, and long-range amplification due to the enzyme engineering, enhanced specificity, sensitivity, yield, and benchtop stability again from its hot start technology. The left figure comparing fidelity values of DNA polymerases in the market, measured by next-generation sequencing. The values were normalized to Taq DNA polymerase. The measurements were made side by side for accurate comparison along with the enzymes listed. And the performance of the platinum superfy 2 enzyme is out of proportion. The right figure shows PCR results with universal annealing temperature of 6 degrees Celsius. It takes away the hassle of having to use TM calculators with previous version of the enzyme. Additional benefits of the platinum superfy 2 DNA polymerase includes higher success with long-range amplification compared to other enzymes, up to 20 kilobases with human gDNA and up to 40 kilobases with E. coli gDNA. Benchtop stability of assembled reactions for up to 24 hours. It is great for high-throughput applications using automation or robotic platform. Innovative buffer formulation allows amplification of targets with over 65% of GC without the need for GC enhancers. Now, turning to direct PCR, which most of you may not be familiar, the conventional PCR workflow involves DNA purification prior to PCR, which can take 30 to 60 minutes depending on the method. Now, direct PCR approach requires no such DNA purification, which cuts down workflow time in half. From sample to PCR can be done in less than an hour. Benefits: no required gDNA purification helps save sample loss and minimize handling. Small samples tend to get lost during purification process. There are fewer steps to PCR and you get PCR results sooner as gDNA purification is not required. Both these translate to more sample processing in a given amount of time. It gives an increased PCR throughput. If you need to purify DNA for applications like restriction digestion, cloning, or DNA assay, direct PCR may not be right for you. The applications for direct PCR, some examples here are animal genotyping and CRISPR-Cas9 screening by PCR are two main applications. Confirmation or detection of target DNA includes samples like blood or buccal swabs, and colony screening in cloning and gene editing. Benefits of platinum direct PCR universal master mix include a single master mix that works with a variety of samples, non-genomic purification required prior to PCR, a single annealing temperature of 60 degrees Celsius that works for primers of differing melting temperatures, therefore simplify optimization. Universal annealing buffer enables universal PCR protocol with one annealing temperature, one extension time, allowing co-cycling. And its formulation with platinum 2 Taq hot start DNA polymerases offers high yield, specificity, sensitivity, and robustness that you can expect from the enzyme.
What is the processivity of a DNA polymerase? Your choices are affinity to the template, number of nucleotides incorporated in a single binding event, rate of DNA synthesis, activation of enzyme activities at a high temperature, or inhibition of enzyme activity at low temperature. Give you a few seconds to answer.
Correct answer for this one is our letter B. Nucleotides incorporated in a single binding event.
Would you like to have a quote of the platinum superphi 2 DNA polymerase? Yes or no? Thank you.
We've now got the DNA template, the DNA polymerase, the reaction buffer, and the NTPs. The last component that's missing are the primers, which can be the forward and reverse primer that specify the target sequence to be amplified. If primers were not designed correctly, you will either get no amplification or lots of non-specific products. It is essential to follow primer design rules to design primer for optimal results. Here in the middle, you can see several tips to keep in mind when designing primers. Primer length, GC content, number of dinucleotides or repetitive bases, these are just some of the aspects to consider when creating a primer pair. Or you can use specialized tools for primer design where you do not really need to think about these rules. One example would be the OligoPerfect Primer Design online tool. At Thermo Fisher Scientific, we have over 27 years of experience in providing a complete range of custom synthesized oligonucleotide primers, probes, and genes, all built to your specifications. Support on-site custom audits for supplier validations. Oligos undergo analysis by mass spectrometry and HPLC to confirm and identify the final purity. And also, they are delivered with certificate of analysis. One more essential component of every PCR reaction is PCR plastics, PCR tubes or plates. Choosing the right plastic consumables is crucial for optimal PCR and qPCR performance. It is important to consider the instrumentation, volume, and extra features when selecting plastics. Different plate formats are available based on the cycler compatibility and automation requirements, such as semi or fully skirted plates for robotics and non-skirted plates for flexibility across different instruments. Low-profile plastics are recommended for reducing evaporation and enhancing thermal conductivity. The volume size for 96 well plates is either 0.2 milliliter or 0.1 milliliter, while 384 well plates are 0.02 milliliters. Additionally, customization options such as barcoding and automation compatibility should also be considered. The right plastic selection can significantly impact PCR and qPCR performance and should not be overlooked. Let's not forget seals. The two most common types of PCR seals are cap strips and adhesive films. Cap strips may be used to seal both PCR and qPCR tube strips and plates, but films are designed to seal qPCR and PCR plates. You can also find PCR and qPCR plastic consumables compatible with thermocyclers and real-time PCR instruments from any company at thermofisher.com/findplastics. Once the instrument is chosen in the Finding Plastics tool, it shows the block types available, and based on the plate format, the tools display also the results showcasing key elements to enhancements like capability, barcoding, automation, along with add to cart feature. There's also an optional feature of download and share, and lastly, one of the most important features, stock availability function is available before cart checkout. Now, with the DNA template, DNA polymerase, buffer, DNTPs, and primers, the reaction setup is ready to go into the thermocycler. However, it is also good practice to set up a few control reactions along with your samples. There are two types, positive controls and negative controls. They serve as an insurance that you can trust your positive as well as negative results. But besides that, they're also extremely valuable for troubleshooting any PCR issues. Positive controls are essential to ensuring the validity and reliability of PCR results. These controls should produce a signal or band on the gel to confirm that the PCR is working properly. The template used for positive controls should have sufficient amount, should not be challenging template, and should be free from any PCR inhibitors. Exogenous positive controls are added to the reaction separately, while endogenous positive controls do not require an additional template as the target sequence is automatically added with the sample template. Endogenous positive controls include a gene, cDNA that should be present in all samples at the same level, allowing for the same cycling protocol, genomic DNA of any other gene with high homology in all samples, or cDNA such as mRNA of a housekeeping gene. Then there are negative controls. Here, every component necessary for PCR is added except for the template. Accordingly, without any template present, there shouldn't be any amplification, of course, so they run to check for unwanted contamination with the template. However, some of these negative controls are positive, meaning some DNA template is present in the reaction. So how did it end up in a reaction? Well, some cross-contamination can happen from other samples present. For example, a DNA template may be introduced because some of the reagent like water or reaction buffer used are contaminated with gDNA or PCR product. So, just to know when this might be the case, it's good to always include a negative control. Otherwise, one can really trust the positive controls. Now, one issue that I come across at Thermo Fisher Scientific that people ask a lot, fairly often, is trouble with the negative control if the assay is designed to detect bacterial DNA. The source here then is usually the DNA polymerase. Most of them are recombinant enzymes, which means that they are produced by expressing them in E. coli and then purified from these bacteria. Unfortunately, it's impossible to remove the bacterial DNA completely from the DNA polymerase prep because of the high affinity between these two. So there will always be certain background of bacterial DNA. To avoid this, you can use a native tag, for example, from Thermos Aquaticus. There are a couple of common guidelines and recommendations to avoid such positive signals in a negative control in the first place. Such, for example, doing each step in a separate place or room, extracting and purifying DNA in one place, then set up the PCR in a second place, run it in a third, and finally do the analysis such as running the agarose gel in a different place. Also, in order to avoid any carryover between reactions via the pipettes, it is common to use filter tips. Also, aliquoting the reagents such as water, DNTPs, and primers help to limit the possibility of contamination. It is then easier to trace back where the contamination occurred and eliminate it. If all of the above fails, sometimes it just helps to make clean start and exchange all the reagents used. Okay, with all samples and control reactions set up, we are ready to go ahead to the second step, the PCR cycling. PCR cycling consists of three essential steps: denaturation, annealing, and extension. In the denaturation step, DNA strands are separated, while in the annealing step, the DNA primers bind to the single-stranded DNA. The third step, extension, involves the extension of primers by the DNA polymerase, copying the complementary strand. These steps are repeated in cycles to achieve amplification. So, the theoretical amount of template DNA doubles every cycle. The multiplication factor is typically around 1.6 due to factors such as pyrophosphate accumulation, degradation of DNTPs, and rehybridization of the template. Now, let's have a closer look at each step and start with the first one, the denaturation. It is important that this step works efficiently so that much primer as possible can bind to the single strands. This process is actually fast and few time is needed to separate the DNA strands. However, depending on your PCR cycler, it takes a few moments until the temperature of 95 degrees Celsius is reached, and a few more until that is also the case inside the reaction vial. The general guideline is to do as long as necessary, but also as short as possible, as the high temperature damages all components, like denaturing the DNA polymerase, degrading the DNTPs, etc. Within the whole cycling program, there are two types of denaturation steps: the initial denaturation, which happens just once, and the cyclic denaturation as a part of every cycle. The initial denaturation often needs to be longer, essentially when it's also used to activate the hot start enzyme. The general recommendation for cyclic denaturation is 30 to 60 seconds, but it depends on the polymerase used and in your DNA template. A few additives like glycerol, DMSO, and such can be added to make this more efficient and if necessary. Annealing step follows denaturation in PCR. Temperature is lowered to allow primers to bind to the single-strand DNA template. TA annealing temperature is usually 5 degrees Celsius lower than the melting temperature, but it depends on the polymerase and primer length. The lowest TM of the two primers is considered. Actual TA might differ from predicted values, so it is recommended to run temperature assay to optimize this step. A gradient PCR is often needed to determine the optimal TA. Calculated TA may provide good results, but an increase of 2 to 4 degrees Celsius may improve specificity further. We already discussed several benefits of universal primer annealing, but another additional and very important benefit of universal primer annealing is that it enables different length fragment co-cycling. Platinum 2-Taq hot start DNA polymerase allows different PCR assays to be cycled together, using the same protocol with universal primer annealing temperature and extension step selected for the longest fragment to be amplified. So now instead of running the thermocycler 4 times, all experiments can be just done in one run. Once the primers have annealed, they serve as the starting point for the DNA polymerase and are extended so that the complementary strand is formed. In order to do this over the required distance, the DNA polymerase needs some time. How much time is needed? That depends on the speed of the polymerase used. Here's some examples. The standard Taq is fairly fast and can do 1 kilobase in 30 to 60 seconds. As mentioned earlier, Platinum 2-Taq and Superfy 2 are much faster and need for the same amplicon size just about 15 to 30 seconds. So one needs to check the speed of the polymerase and calculate the time needed to complete the amplification. Otherwise, if the time is too short, the fragment size will not amplify completely and there will be no amplicon at all. Too long, the overextended products can cause some smear, showing fragments with a range of sizes depending on how far the polymerase has got. These 3 steps of denaturation, annealing, and extension are now repeated several times. 30 cycles are sufficient to give a visible band on the agarose gel or enough PCR product to work with. If the starting amount is very low, it can help to increase this to 40 or more cycles. However, with an increasing numbers of cycles, also there will be an increasing numbers of the changes of misannealed primers and also unspecific products. We see often results where there is some signal close to 50 cycles, but usually it turns out that's just some unspecific product. Less than 25 cycles is likely to result in not enough PCR product unless the amount of starting material was already high.
Would you like to have a demo of a PCR instrument? Yes or no? And thank you for participating in our poll.
This is it, the setup of PCR reaction and thermal cycling. Once the PCR has been run, the result is often analyzed by running the samples on agarose gel to visualize the bands and check for amplicon size. Choosing the right tools for nucleic acid electrophoresis can significantly improve and accelerate results, enabling you to address downstream applications sooner. Determining the appropriate gel type and gel concentration is an essential step that will help streamline the separation of nucleic acids. The eGel precast gels are self-contained and ready to use with agarose, electrodes, and the DNA stain packaged inside a disposable cassette. There are no gels to pour, buffers to make, staining or destaining steps to perform, or gel boxes to assemble. It's very simple. You just load your samples and run in one of the power snaps that you see in the pictures on the right. Can be either the Invitrogen eGel Power Snap electrophoresis system for 25 or less samples at a time, or the Invitrogen eGel Power Snap Plus electrophoresis system for 25 samples or more at a time, up to 96. In both, you can go from sample loading to image capture in just 18 minutes. If the product is needed for cloning, the PCR product is cleaned up and later sequenced as well to check for errors. So there are many roads that lead from here, as PCR is a part of quite a variety of workflows in molecular biology. The method has evolved enormously since its invention about 40 years ago, thanks to faster and more robust DNA polymerases like the Platinum 2 Tech and Platinum Superfi 2. Since they are more tolerant to contaminating substances, it's possible now to skip whole parts like extraction and purification from some PCR workflows and run a direct PCR. While universal primer annealing simplifies PCR preparation and eliminates the need to calculate annealing temperatures. But even in these cases, the underlying principles are still valid. We still need to have template, DNA polymerase, buffer, DNTPs, and primers present in that mix for a direct PCR. And the following cycling still consists of 3 steps: denaturation, annealing, and extension. It might be possible to run such a PCR without knowing too much about what's going inside the vial. But if you do, it's much easier to develop a new assay, or even push the method beyond its current boundaries. Now, let's take a look at some resources. The product information pages, such as for PCR enzymes, thermal cyclers, and PCR plastics, lets you choose the best product suited for your application, based on properties, experimental needs, and formats available. If you're interested, we also have a plethora of application notes. The PCR workflow solutions provide you detailed information and many supporting data as well of many of the products that I touched base today. Our mobile school and PCR education sites provides you mobile basics, tips, troubleshooting guides for new and experienced researchers alike. Their respective URLs are provided under each of them. Also, at the bottom part shows key brochures and handbooks related to PCR workflow. You can even find an interactive version of the handbook with videos and valuable information incorporated into the handbook. Please check it out, all these resources. Drive your lab efficiency with powerful PCR tools designed for speed, simplicity, and accuracy. This was our webinar today. Thank you so much for watching.
Dr. Alves, thank you for your time today and for your important research. And before we go, I want to thank our audience for joining us today. I'd also like to thank our sponsor, Thermo Fisher Scientific, for sponsoring today's webinar. Questions submitted today and during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand for 2 years until March 22nd, 2025. Laboratories will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Bye-bye.